I'm live. This is actually going to work. I'm hotspotting my internet through my phone on Verizon because I don't actually have real internet out here in the woods. Yeah, it's pretty amazing. You would think that we can get internet, but satellite hasn't worked. So I'm really glad I can be here today to live stream with you guys. So this is for both 303, which is the Ecology and Evolution course, and 304, which is the Plant, Animal, Form, and Function. And both these classes this summer, I've got both of them going on at the same time, and I've made everything in both classes basically the same. You've got a test once a week. Each test has got 10 multiple choice questions with 5 essay questions. 4 of those are worth 5 points, and 1 is worth 10 points. And so it's been kind of interesting getting both these classes kind of on that exact same format. So what I want to do now that we're live is I want to take on any of your questions that you may have, and just you can live chat with me. And I'm also going to answer some of the questions that some of you have sent to me through email, because I'm getting a couple really good questions that you guys have been emailing me, and I've responded to some of you personally. And then there's a couple that I was like, well, I'm going to get to it today. Okay, so let me get situated here, not bump my table too much. But one questions that I had was from Plant and Animal Form and Function about alternation of generations. Now, plants they're different from us, right? I mean, you and I, we're a diploid organism. We've got 2 copies of our genome, one from our mom, one from our dad. So we have 46 total chromosomes. 23 of those are from one parent, 23 from the next. And then you and I, we form gametes, either a sperm or an egg, that are haploid, that are made through meiosis, which is a type of cell division. Let me turn my radio down here. That cuts the number of chromosomes in half. And then as an adult, all of our cells are constantly reproducing by mitosis, which produces somatic cells, or cells of our body, that are also diploid. Now, here's how plants are really different. Okay, plants, especially like little mosses and things like that, these plants are they have what's called alternation of generation. So they have 2 different body forms. They've got a sporophyte and a gametophyte, and that's pretty awesome, right? And then I'm just double checking something here really quick. With that, oh, I lost. Yes, okay. So the sporophyte. Here's what's interesting. The sporophyte is diploid. Okay, that means they've got 2 copies of their genome, and the sporophyte will create these things called spores through meiosis. So that means that the spores created by a sporophyte through meiosis are haploid. Now, our gametes, they just fuse and form a zygote, which is diploid, and that goes off to become us. But in a plant, especially like mosses and ferns, here's what's interesting: that the spore will actually grow into a haploid organism called the gametophyte. So if you're ever hiking up in the sandias and you see these tiny little greenish little moss-like plants along the trail, or up against some rocks or something, those are almost certainly gametophytes, and they are haploid. And what's interesting is that the gametophytes will then produce gametes by mitosis. Well, I just said that right, didn't I? The gametophytes make their gametes by mitosis because they are already haploid. That's pretty wild, isn't it? Could you imagine, like, you produce a gamete that grows into a whole nother individual? Yeah. And then they produce, these gametes that will then fuse. And then when they fuse together, they become diploid and form the sporophyte. So it's called alternation of generations, because you alternate between a sporophyte, which is a multicellular organism, and a gametophyte, which is awesome. How cool is that? I get really excited thinking about alternation of generation of plants, and that was one of the better questions I had, because that can be quite confusing. I also had another question, and this one goes to Biology 303, which is my Ecology and Evolution class, and somebody was asking me about Lamarck. Now, a lot of biologists, especially when I was in graduate school, we used to make fun of Lamarck. Ha ha ha, Lamarckian biology, Lamarckian evolution. Go giraffe, stretch your neck, reach that plant, and then by stretching its neck all throughout its lifetime, it would somehow be able to pass that on to the next generation, and it was called acquired characteristics. One way of thinking about that is, well, I've been working out since 1989. with weights right up until March. And because I've worked out my entire life, I got stronger, and maybe somehow that would be passed on to my children, or my child, actually. No, by the way, yeah, wife is due in November. Woohoo, got number 2 on the way. But just because I work out doesn't necessarily mean that my child would be stronger because I worked out, that I acquired this characteristic of strength that would be passed on. So Lamarck, we often made fun of his acquired characteristics, that whatever happens in that animal's life is somehow passed on to the offspring. But one thing that I really like about Lamarck that people forget, Lamarck came along in the 18 teens, like, I don't know, 40, 50 years before Darwin published on the origin of species. And, interestingly, he proposed a testable hypothesis for evolution. Lamarck thought that species evolve over time, and he actually provided the mechanism, which is acquired characteristics. But, once we started understanding heredity, beginning with Gregor Mendel and on to Thomas Hunt Morgan, and then all those guys of the modern synthesis in the 1920s, 30s, and into the 40s, we realized that's not exactly how evolution works. It is natural selection by passing on genes to the next generation. So acquired characteristics was a testable hypothesis. It was one of the very first testable hypotheses of evolution, but it's not the way evolution works until now. Now we know that there's this thing called epigenetic inheritance. And epigenetic inheritance kind of is Lamarckian, which is like throwing us off a little bit. Epigenetics, epi means above genetics. And what happens in your life, you can actually pass on to your children and your grandchildren, but it has to do with the way that genes are regulated versus a natural change in the DNA. So we don't know like how long epigenetics will last, for how many generations. We do know it goes on for at least 2 generations, and that could affect natural selection. And of course, we learned about epigenetic inheritance from things like the Dutch hunger winter. And that's kind of an interesting thing. In the 1940s, the Dutch sided with the Allies, and the Germans got mad. And what they did is they shut down all roads into the Netherlands, right? And they cut off their food supply, and they starved. It's called the Dutch hunger winter in 1945. And when they starved, people went on about 30% of their rations. Well, what they discovered is that women that were pregnant before the hunger winter but gave birth during the hunger winter gave birth to smaller babies. That makes sense because babies gain most of their weight in the 3rd trimester. But those children stayed small their entire life, and then their children also stayed smaller. On the side, women that got pregnant during the hunger winter and were starving, but then gave birth afterwards, gave birth to normal weight babies. But those babies grew into adults that had problems with obesity, and then their children had problems with obesity. And the reason why was that starving early on in the development, the first 2 or 3 or the first 2 trimesters, was enough to change the way that part of the genes involved with metabolism were being regulated. So that's pretty wild that what does happen to the parents can be passed on to the offspring and be there for a couple generations. It's just we're not actually changing the DNA. Okay, so those were 2 of the main questions that I received over the last week or so about ecology and evolution and plant and animal form and function. So if anybody has if you've got any questions, you can live chat me right here. If not, I got some other things to talk about. And it's kind of interesting. There's always so much to talk about, but I won't waste your time too much. I want to go to 304, plant animal form and function. And right now, you guys are studying transport in the xylem and in the phloem. And then in the next chapter, you're going to get into plant nutrient uptake. And then in the second half of the summer, we're going to talk about animal form and function and homeostasis. And there's one connecting thread that I want you guys to really focus on. And this goes back to cell biology, and that is transport mechanisms. And transport mechanisms include, facilitated diffusion, like you're using ion channels, and also active transport that involves both primary and secondary active transport. Primary active transport is like a sodium-potassium pump. I use ATP. I'm going to pump sodium out of the cell. I'm going to move potassium in. That would be an example of primary active transport. A proton pump, think like, oh gosh, cellular respiration, there's electron transport chain pumping protons into the intermembrane space. That would be active transport, and we also call that primary active transport. Now in plants, to move things like to move sugars into their phloem, they use primary and secondary active transport. So if you want to get sugars from like the mesophyll cells where there's photosynthesis taking place, right, and you got to get them into the companion cells, stuff them in there, right? So what the plants do is it's pretty cool. They pump protons out of their cells, right, and then they use that proton to go back down as concentration gradient. And while they do that, they drag in a sucrose molecule with it. And it's called a symporter because the protons and the sucrose are going in the exact same direction, right? So you're pulling the sucrose against its concentration gradient, but you're using the proton going with this concentration gradient. And that's a secondary active transport. It's because the primary is a proton pump, then the secondary is this dragging the sucrose molecule in there with it. And that's through a symporter. And then when you unload the sugars and you want to get them into a vacuole, use an antiporter. So in this case, the vacuole will pump out a proton, and then it'll come back in, and then the sucrose molecule will go in the opposite way of the proton, right? So that's called an antiporter, and they're both secondary active transport. And then when you get into, well, how do plants move nutrients into their roots? Well, guess what they do? They actually pump protons out of their roots, and that creates, one more time, an electrochemical gradient. You got an electrical gradient because the protons are positively charged, so the outside the cell becomes really positively charged, and then the inside of the cell becomes really negatively charged. All right, we got an electrical gradient, and it's a chemical gradient because there's more protons on the outside than there on the inside. Combine that together, we call that an electrochemical gradient. And because there's a charge across that membrane, we're going to call that also a membrane potential. These words are lots of words in the book, but I'll use the electrochemical gradient. And then, so what happens when you get cations, which are positively charged ions, think like potassium, calcium. What they do is they'll pump the protons out of the root, and that proton will then knock these things off the clay particles. It's called cation exchange. But because you've created a negatively charged interior, what will happen is that negatively charged interior will it'll pull the potassium ions in through a potassium ion channel. Facilitated diffusion, right? Remember that from cell biology? That's facilitated diffusion. So they're creating this electrochemical gradient, but then they move the cations into the cell by literally pulling them in with a negative charge. And as long as the electrical gradient is strong enough, it will move the potassium against this chemical gradient. And then if you're an anion, which are things like phosphates and nitrates and chloride ions, what they'll do there is they'll do secondary active transport. You pump the protons out of the cell, the cells come in. The protons come back in through the cell, and they'll come in through a symporter dragging these things with it. So once again, there's active primary transport, secondary active transport to move these anions in. So you start to see how this transport phenomena is very, very important for plants. And then when we get to animals, we're going to find out that the way we maintain homeostasis is also by using active transport, both primary and secondary. So there's this connection between plants and animals that we're both having to do these similar things. Okay, I got a question. Hi, Tom. Does the number of stomata on needles and leaves of a tree influence flow rate of water up the xylem, and how does the number of stomata and angiosperms compare with gymnosperms? Wow, Lucas, you actually got me a little bit on how they compare to angiosperms and gymnosperms. Remember, angiosperms, for those of you, these are flowering plants. So think like a rose, an oak tree, grasses. These are, even the grasses have lost their flowers. Those are the flowering plants. And then gymnosperms are things like pine trees and conifers, fir trees, cedar trees, stuff like that. And if you remember, gymnosperms they have these needle-like leaves, and then the stomata are embedded in those leaves and deep. And there's a couple ways that they do affect the rate of water movement. There's a couple of things that do affect the rate of water movement through these plants and how they can conserve it. So I can answer that because that's what's going to be on the test. So sure, if you've got a lot of stomata on leaves and needles, you can theoretically have water flow. And of course, you open up the stomata, and that allows evapotranspiration to occur through the cohesion tension theory, where it pulls water up, like sucking water up through the straw. It sucks water up through the roots, through the xylem, and out the leaves. Ways that plants conserve this are you close your stomata during the day, and that's actually involved with some chemical signaling and the movement of potassium in and out of those leaves, which affects the water potential, which affects the shape of the stomata. So during the day, they open up, and then when they get water stressed, the stomata will close, and that will stop the flow of water in there. Also, in the underside of leaves, if your stomata are like way up in the cuticle and there's all these little trichomes around it, that will increase the humidity right around the stomata. And by putting a lot of trichomes right around it, if you ever look at a leaf that's hairy on the underside, that's actually creating a little tiny microclimate in there, making it more humid, which is actually keeping the water potential less different than it is inside the leaf, and that will slow down the rate of evaporation. Also, the xylem is made up of both sieve tubes and vessel elements, right? And in gymnosperms, they only have one of them, right? And they're much more small and narrow, and they're missing the perforations at the top, whereas the angiosperms have both. And the angiosperms, because they've got the perforations in the top of their elements for the xylem, they can actually move more water more quickly than a conifer. And we think that's part of the reason why angiosperms, why there's like 230,000 species of angiosperms, and they're so incredibly abundant or diverse compared to conifers. Like there's only 640 species of pine trees, right? And so we think that ability to move more water more quickly is one of the reasons why angiosperms have become way more diverse. But there's always trade-offs, right? The largest forests in the world are these northern boreal forests that just blanket North America, Europe, and all across Russia and Asia. And even down here in New Mexico, we have lots of these coniferous forests because they can do much better in cooler and drier environments. Okay, so Lucas, that was a good question. I hope I kind of answered it. I know I answered it in the way I would ask it on a test. Anybody else have any more questions? Okay, there's one more thing in ecology and evolution that I want to bring up for you guys in 303 out there. One of the things that the book really it's a good book. I mean, it's huge. It's a tome of knowledge. But it's not the end all to be all to biology. That's why I'm giving you guys a lot more information than what's in the book. And one of that sources of information I want you guys to work on is that modern synthesis and understanding like the role of understanding genetics and heredity and evolution, and also how traits are passed down, and the population thinking that like populations are the smallest thing that can survive. I mean, that can evolve. And then of course Ernst Mayer's definition of a species. Because one of the things when you get into the next week about origin of species, one of the most ironic things about Darwin is that in his book on the origin of species, guess what? He doesn't really talk about the origin of species. He talks about evolution by natural selection and presents a lifetime of evidence explaining or providing evidence for natural selection and that species change over time. But in Darwin's time and right up until like the 1940s, and when Ernst Mayr came up with a biological species definition, we didn't have a good example of what a species was. And his, of course, this reproductive isolation really helped out because you could now think about allopatric speciation and things like that. But we now had our first definition of a species. Now that we know what a species is, now that we know what the unit of evolution is, natural selection, and natural selection is acting on the individual, based on this combination of alleles, right? That those individuals that are more likely to survive are more likely to reproduce and pass on those beneficial alleles, and they amplify in a population over time. So that's important to make these connections in the modern synthesis, which is something I'm going to talk about next week. I'm actually going to do, I think, a little podcast on that. And like I said, if you guys have any suggestions for podcasts or anything I can do to help get this material out there better, you just let me know, because I'm always open for suggestions, and I'm very excited that I can do these live chats with you guys. And I'm also very flexible on my time and days to do this as well. Well, I don't have any other questions, so I'm gonna sign off. Lucas, man, that was a great question. And anybody else, if you got any more questions, now's the time. If not, I'll sign off here, and you can always hit me up on email and ask questions, because like I said, it's kind of fun, when you guys ask me questions, but on email, tell me what you're thinking. Don't just say, hey, Tom, I don't understand this. Tell me what you're thinking, and then that way we can start up a dialogue about it, and then I can also come on here and explain it further too. All right, well, have a great weekend. Don't forget, test 1 and 2 are both due Sunday night, and I'm going to put up our first set of essay questions, and I hope the essay questions are actually kind of fun. I don't want you to really fret over them, worry about them. I want you to think about the answers, and this should be a thing that's kind of fun to think about. Okay, I'll see you next week. Bye.